these will be our questions. Let's get into it, guys. I'm trying to do this really, really quickly because I want your revision to be efficient as well. So, organelle or structure, appearance, because a lot of this is visual, right? You'll get a picture of an organelle, you'll have to identify it, you'll have to identify its structures, and function. And you'll have to appreciate, at least at a superficial level, like how does it contribute to the rest of the cell. Nucleus, appearance, okay? It's the big organelle in the center of the cell, stains darkly with DNA stains, okay? So, nucleus, function, contains chromosomes, right? It contains chromosomes which have the genes, or chromosomes are made up of many genes, and the genes ultimately made up of DNA. Okay, good. And ultimately, these genes are the instructions to make proteins, but we won't spend too much time there. Okay, then we've got the nucleolus, right? I don't know how quickly I can do this. So if we have a cell, then we have a nucleus, and in the nucleus you've got these dark bits. That is our nucleolus, or what's its purpose, the nucleolus. There's lots of transcription going on there. That is where the ribosomes are made. Yes, the ribosomes are actually made in the nucleolus, and then the ribosomes make their way out into the cytoplasm and get attached to the endoplasmic reticulum. All right, we also have, I guess we could attach it, or maybe we can have two sections here. We can have the nuclear membrane, or the nuclear envelope. So that's this part right here. So the nucleolus are the dark staining bits in the nucleus, and the nuclear membrane, well, that's the membrane that forms the boundary of the nucleus and separates the nucleus from the cytoplasm. Okay, and the nuclear membrane, I guess it compartmentalizes the DNA away from all the other cell components that could cause damage to the DNA, but it also contains pores that allow things to go in and out of the nucleus as per the requirements of the cell. So when an mRNA is made in the nucleus, the nuclear pores allow the mRNA to come out. Okay? Or if there's transcription factors out here, it allows them to go into the nucleus and affect which genes are being transcribed. Okay? So I'll just put down nuclear pores for things like being able to go in and out of the cell, and we can say also that it protects the DNA, the chromosomes. Okay, next we have our endoplasmic reticulums, rough and smooth. Okay, so the rough endoplasmic reticulum, RER, and its appearance is roughly, okay, so it's attached to the nucleus in a way, but it's interconnected, flattened sacs. Yeah, interconnected, flattened sacs. So they're made of membrane, but they have ribosomes attached, like so. And remember, the role of ribosomes is protein synthesis, so the function of the rough endoplasmic reticulum is also protein synthesis. They bind messenger RNA and use the instructions of a messenger RNA that came out through the nuclear pores, that was made in the nucleus. They bind to that mRNA, the protein is made inside the cisternae of the rough endoplasmic reticulum. Okay, right, so protein synthesis, yeah, okay, and we'll leave it there. There is also a smooth endoplasmic reticulum. All right, now it looks a little bit different. It's also attached to the nucleus, it's also made of membrane, but it is more, they're interconnected, but they're not flattened sacs. So they are connected in a way, but they are not flattened sacs. And the role of the smooth endoplasmic reticulum is more to do with lipid and carbohydrate synthesis. Okay, so lipids, we're thinking about phospholipids that make up membranes. Carbohydrates, we're thinking about the carbohydrates that get attached to proteins that form the glycoproteins in membranes. Okay. Now, yeah. Okay. Let's just keep moving on, shall we? Smooth endoplasmic, rough endoplasmic tube. I guess we should mention our little ribosome at this point. Okay. Now ribosomes are mainly RNA molecules, so they're RNA and protein combined, but they're mainly made of ribosomal RNA. That's why they're made in the nucleus. But their job is to use an mRNA molecule as an instruction to join amino acids together to make proteins. Okay, and they kind of have like a hamburger kind of structure. They've got a big portion and a small subunit, and they attach together to make the complete ribosome. Okay, once a protein is made in the rough endoplasmic reticulum, it will then get packaged up. Not packaged up, but well, it will be packed into a transport vesicle. So transport vesicles are a thing, so let's just call them vesicles. Vesicles are just membrane components, and they contain things, and they are taking things that originated in one organelle and take it to another organelle, or they might take it to the cell membrane to be ejected from the cell. Okay, so vesicles, they have their membrane part and they have their contents part as well. And there are different types of vesicles. So you have transport vesicles, and you have secretory. So transport vesicles take things from the rough endoplasmic reticulum to the Golgi apparatus, and the secretory vesicles take stuff from the Golgi apparatus to the cell membrane. Okay, transport and secretory. Okay, I guess that then takes us to Golgi apparatus, Golgi body for short, I guess. Right, and that is the thing that looks like that. It's got flattened sacs, but they're not interconnected. Okay, and what will happen is they will receive, so one of these transport vesicles from the rough endoplasmic reticulum will bring proteins to the Golgi apparatus. The Golgi apparatus will modify those proteins, it will stick bits onto the protein, it will modify those proteins, and when those proteins are ready to work, at the other side of the Golgi apparatus, they'll be packaged into a secretory vesicle, or a vesicle that's going to another organelle, its final destination. Okay, so Golgi apparatus modifies and packages proteins. I might add, though, there's also the cytoskeleton, and part of the cytoskeleton is the microtubules. Now, microtubules are long, very rigid, kind of fibrous arrangements of the protein tubulin, so they form these filaments. Now, microtubules, they are the network on which the vesicles are moved around the cell. So when a vesicle moves from the rough endoplasmic reticulum to the Golgi apparatus, it moves along the microtubules. So one function of the microtubules is intracellular transport. And in fact, all the organelles are kind of stationed where they are thanks to the scaffolding provided by the microtubules. What are these other organelles then? Now, lysosomes are kind of connected to the protein synthesis story, because many proteins which are made in the rough endoplasmic reticulum are digestive enzymes. And those digestive enzymes are stored in the lysosome because otherwise they're too damaging to just keep lying around in the cell, because they'd start digesting all the cell materials or components. So these lysosomes are basically just glorified vesicles, but their contents are what make them unique. They have the digestive enzymes inside. So whenever the cell wants to break anything down, the cell sends it to a lysosome to get broken down. And whenever there's phagocytosis happening, the thing that has been ingested also gets sent to the lysosome. Okay, so digestive enzymes are in there, and that's part of its structure, but its function is kind of breakdown of cell components, but also it's involved in digesting pathogens, right? So if there's anything pathogeny that's been ingested from, by a white blood cell, for example, once it's in the cell, it needs to be sent to the lysosome so the enzymes in the lysosome can break that stuff down. Okay? And these digestive enzymes, they were delivered to the lysosome by a vesicle from the Golgi apparatus. So if you're looking for connected stories, that's how you do that. Mitochondria, right? Now, for most of the things that the cell does, if it needs energy, it's going to get that from the mitochondria. Okay, so these are quite large organelles. They have an outer membrane, they have an inner membrane which is folded to increase its surface area. Okay, and these sticky out bits are called cristae, and there are also these particles called stalked particles, which are very important in ATP synthesis. Okay, so outer membrane, inner membrane, you've got the intermembrane space in the middle, and then in the very middle, within the inner membrane, you've got the matrix. Okay, you've also got these cristae, and you've got these stalked particles as well. But basically, the mitochondria are involved in aerobic respiration, and its main contribution to the cell is it gives the cell the ATP that the cell needs to do all these other things like protein synthesis, okay, or phagocytosis, or cells moving around. Right, then we have chloroplasts. Right, these, I think, maybe after the nucleus, these are the biggest organelles. So you've got chloroplasts. Chloroplasts are very big. Outer membrane. Inner membrane, yes, you should be able to sketch these out. I mean, yeah, the diagrams look detailed, but they can be simplified. Right, outer membrane, inner membrane, then you've got this fluid inside, which is called the stroma. Then within that fluid, you've got other folded membrane structures called thylakoid membranes or thylakoids, and this is where the photosynthesis happens. Okay, again, it's folded slightly in a different way to mitochondria, but it's folded to increase the surface area, and it's these thylakoids that have the chlorophyll that does, that captures the light energy to make glucose from carbon dioxide. Okay, so their main purpose is photosynthesis, right? And remember that while aerobic respiration, we're basically using glucose to make ATP, photosynthesis's purpose is to make glucose out of carbon dioxide. That structure is also important. The cell membrane or the plasma membrane separates inside from outside. And remember that, it sounds basic, but the reason for separating inside from outside is that the inside has got more chance of being controlled, and the contents can then be more amenable to serve the functions of the cell. Separates inside from outside, defines the boundary of the cell, whatever you want to call it. Okay, made of phospholipids, has got proteins, transport proteins, etc. And we look at more of the structure of the membrane when we do biological membranes. So we've got plasma membrane. I think then that takes us to cytoskeletal components. Okay, now here I'm going to divide this section into three. Okay, because there's three main cytoskeletal components. There's microtubules, there is microfilaments, and there are intermediate filaments. Okay, now microtubules form a number of different structures. So microtubules form flagella. Microtubules form cilia, right? And microtubules form centrioles. Okay, but they also form that network that we talked about for transport. So can I just make another one here? Microtubule network. Okay, rough and ready. It's messy, but this is how we revise. This is not how we revise, this is how we retrieve. Okay, and it's not going to be pretty all the time. So microtubules, flagella. Okay, so flagella, purpose, motility, right? It moves the cell. Cilia can have two functions. Cilia can be motile cilia, but that's not to move the cell, it's mainly to move fluid over the cell, right? Like the ciliated epithelium in the lungs. So we've got motile cilia, but we've also got sensory cilia. So some cilia are just there to detect kind of vibrations in the air. Centrioles form the spindle fiber and aid in chromosomal separation during mitosis and meiosis. And the network of microtubules that goes throughout the cell, that's for intracellular transport, i.e. the transport that happens within the cell. Microfilaments, they change the shape of the cell. Now, when does that happen? Well, the cell shape is changed when a cell moves. When a white blood cell moves from the blood vessel towards the site of infection, that is happening because of the work of microfilaments. When a cell moves, and also when a cell does cytokinesis. When a cell does cytokinesis, when one cell splits to become two cells, that's thanks to the work of microfilaments as well. Intermediate filaments, pretty boring, but basically they provide the basic cell shape, and they're there to just support the cell, give it structural integrity. So it's more about strength and shape. Okay, microfilaments, it's to do with changing cell shape. They're very thin, right? So microtubules, they're very thick. They're the thickest. Microfilaments are the thinnest, and intermediate filaments are somewhere between the two. And I'm sure I've forgotten some stuff. One last thing would be cell wall. Okay, now the cell wall that we need to know about in detail is the cellulose cell wall, but the cellulose cell wall is in plants, right? And it gives support to the plant cells. But there's also a cell wall that's chitin, which fungi have. They are also eukaryotic. But remember that there's also the peptidoglycan cell wall of the prokaryotes, which is again different, but though their functions are similar. Okay, and I think we'll leave it there. So that's us kind of just quickly going through all the organelles. Our third and final question is going to be, what are the main differences and similarities between eukaryotic cells and prokaryotic cells? And that should take us to the end of cell structure. Now, in terms of the DNA, what are the similarities? Well, ultimately, the DNA is the same bases, same overall structure, same DNA double helix structure. Yeah, but in terms of differences, quite a lot. The DNA in the eukaryotes, so in eukaryotes, it's in a nucleus. In eukaryotes, it's linear, right? Whereas in prokaryotes, it is circular. Yep. In eukaryotes, the DNA is associated with histones, whereas in prokaryotes, you can consider it to be naked, right? It's not bound to any proteins or as much associated with proteins. And the location, I guess in eukaryotes, the location is a nucleus. In prokaryotes, the location of the DNA is cytoplasm, right? In terms of organelles, similarity, we do have ribosomes, so they both do have ribosomes. But the differences are that the eukaryotic ribosomes are bigger, so you've got 80S ribosomes in eukaryotes and 70S, which is a measure of the size, ribosomes in prokaryotes. In terms of organelles, the difference is that eukaryotes have membrane-bound organelles, Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum, no membrane-bound organelles in the cytoplasm of a prokaryotic cell. None in prokaryotes. Okay, so that's a lot of differences summarized in that. So just be wary of that. Size. Size, they don't really have a lot in common there. Differences in size, eukaryotes, much larger cells. Cell wall similarity is that both can have cell walls. Not animal cells, obviously, but plant cells can. Plant cells, fungi, they have cell wall. However, prokaryotes, they do have a cell wall, but prokaryotic cell wall is made of peptidoglycan. So it's made of different stuff. Okay. Cell membrane, they both have it, so that's a similarity. Both have cell membrane. I guess the difference could be that in a prokaryote, the cell membrane might have different lipids. Yeah, different kinds of lipids that are associated, that are making up the membrane. And in a prokaryote, the membrane is not the outermost structure. Okay, now that's similar to plants and fungi as well. But in animal cells, remember the cell membrane is right on the outside, whereas in prokaryotes it will be a cell wall on the outside. So there's a number of different ways that you could do this. This is just my suggestion, and this is a very quick overview of those similarities and differences. And that brings us to the end of our third question from this aspect. So I'm going to get rid of this. Okay, and we're going to wrap up this little session with a reminder of our three essential retrieval questions. These are the questions that we've got to keep practicing. What are, or how are organelles involved in protein synthesis? How are organelles involved in protein synthesis? What are they? What are their overall structures? What are their contributions to the overall process of protein synthesis? 2, what are the other organelles? Similarities. What are the similarities and differences between prokaryotes and eukaryotes? And we get to work. Okay, so we've just gone over this. I think if you can get stuff down in response to these from memory without any help at all, I think we're getting somewhere. Okay, and I think just making some progress is a sign that, well, much more progress is on the horizon. Okay, but we've got to get things moving, and I think if you can answer these questions, what's to stop you asking the next set of questions? And if you can answer the next set of questions, well, what is stopping you from answering any question in biology, really? All right, so that's the spirit in which we're going to do this. Hope it's been useful. You now get to work. The easy part is done. Let's practice those questions. And what I would suggest, we're now 3 sessions into this thing. Go back and look at some of the first set of questions on microscopes, on comparing microscopes. How many of those questions can you answer? It's going to be a bit trickier to do it now compared to when we just went over it, but over time you'll get better and better. Okay? All right, guys, see you in the next one. Good luck.